Welcome and welcome back to those who have already listened to our pilot podcast of the Pharmaversity last week. Today, you listen to the second edition, which is basically a logic continuation of the previous week's podcast. I finished with an outlook to the EVERIC family, and Florence mentioned the different family members. Here we go. Today, you will learn more about EVERIC strong and smooth two family members. You probably know from last week that I'm not a chemistry expert, and that's why we skipped the chemistry lesson today, and we will talk a lot about efficient processing and machinability topics in this podcast. Obviously, I'm not alone for this. I'm very delighted to have Diana Löber on the line today. She's Global Product Manager for Vials at SCHOTT's Business Unit Pharmaceutical Systems. Ever since, Diana set her sails in the healthcare and pharmaceutical industry. We know each other, Diana, already now for almost 2 years, and I'm very happy that you will be sharing your knowledge on this interesting topic and case study today. Diana, welcome and great to have you here.
Yeah, hi Björn. So this is really on my side to thank you. So I'm really thank you for having me, and that I have the opportunity to talk about our EVERIC vials today.
Great. I want to get straight to the topic, because you told me that you were live at the production line tests at Roche, and I can tell you I want to know all about it, and we come to that later. So let's get started. And my first question is, because I heard one of your recent presentations on the topic, the SCHOTT EVERIC family, and you said it's a modular approach for our customers. Can you explain that a bit for us at the beginning?
Yes, of course. So user centricity is really essential for us at SCHOTT, and our goal is always to offer specific solutions to meet our customers' specific needs. And this is modular concept of EVERIC. We strive to adapt to this new pharma environment and its requirements, providing solutions for drugs of the 21st century. And actually, two main drivers have been behind the development of this modular concept of EVERIC. So one we call the perfect fit for use. And some of you might have heard of it in the last podcast already. And a background here is that 2/3 of all newly development drugs will be biologics. And therefore, we strive to offer solutions for those often big and complex and highly sensitive molecules. So tackling this perfect fit for use, it's all about chemistry, about the potential drug-container interactions and their consequences. And EVERIC Pure is our solution here. An improved borosilicate glass in combination with an improved production technology, leading to this unmatched drug stability and having determination under full control. So this is what EVERIC Pure is. But like Björn said, we do not want to talk about the chemistry part today. So besides this perfect fit for use driver, we have the second driver, and this is what we call efficient processing. And one part of this efficient processing driver is that we want to have a stronger vial, but without changing the glass composition or needing to treat the glass somehow. Because doing so, so if we change the glass itself, this would really have stood in strong contradiction with what we achieved with the EVERIC Pure module and its pristine glass surface. So our solution, or our module for this stronger vial, is what we simply call EVERIC Strong. So EVERIC Strong is all about optimized strengths, realized by an optimization of geometry. And we managed to get there through the analysis via mathematical modeling here. And the second part of this efficient processing driver is EVERIC Smooth. That's all about protecting the vial surface and achieving a smooth fill and finish process by an outer coating. Yeah, so this is the EVERIC family with its different modules.
Okay, I would like to dig deep into the scientific approach of mathematical modeling. That sounds very interesting to me. If I understood it correctly, it's basically used to find the perfect geometry of a glass vial within the converting process. Would you go into a bit more detail for me on what exactly you're doing here?
Yeah, we noticed that when we performed strength tests, we got differing results from different batches. And we simply wanted to find out why is that? So what is so special about the strong vials? And then we used mathematical modeling to find that out. Yeah. And luckily, due to the fact that SCHOTT is a manufacturer of special class, we have a large R&D department with a lot of experts that specialize in that field and have strong competences there. So what we did, we varied the different parameters, and we could see via this mathematical modeling approach that with certain parameter values, there is inherent stress in the vial. And with this inherent stress, I do not mean the stress due to a thermal gradient. This stress we release via our annealing procedure. This inherent stress is based on the geometry itself. And the bigger the stress is, the bigger the likelihood is that the vial might fail under pressure, under load. So stability is all about the right geometry, angles, relation of sizes, and distribution of material. If you, for example, think of a chair, so there are different ways how its legs can be positioned in relation to the seating area with different angles. And this plays a big role when it comes to the stability of the chair. So the idea behind this was to tighter control these parameters of the vial, which we identified as being responsible for those inherent stresses. This means that we did not change the parameters themselves. We stay with an ISO here. It's just about achieving tighter tolerances, a more uniform result, let's say. And again, we are really lucky to have this in-house knowledge regarding machine development. So we could directly transfer our assumptions from theory to practice and produce those vials with an optimized geometry. And again, we used the competences of our R&D center and performed strength tests here. The results of those tests proved then that we truly achieved what we intended to achieve when we started the project. And the product, what came out of this research and development, is EVERIC Strong.
Okay, that sounds really interesting to me, but I don't want to let you off the hook so easily, because everyone could say our vial is stronger, we have done tests and now it works. How can our customers be sure that you actually get what we promise?
Yeah, great that you mentioned this, because this is exactly what we thought about as well. So if I was a customer, I want to have a concrete value here. So instead of just claiming, yeah, our vial is 2 to 3 times stronger or similar marketing slogans, we decided that we will have a statistical in-production release test confirming the strength of the vial. And this test will be based on real absolute values that the tested vial will then have to meet. And this will come in combination with an according AQL level.
Okay. So if we look at a typical fill and finish line of pharmaceutical manufacturers or CMOs in the world, what exactly are the consequences in real life situations if they would use EVERIC Strong vial now?
So we know that glass breakage is something that might occur, but usually only in rare occasions at our customers' filling lines. But if you have this occasion, so if you have glass breakage, it's taken the whole production on hold. And it's a very time-consuming procedure to do the cleanup. So with EVERIC Strong, your risk of breakage is reduced, simply resulting in less downtimes. And as mentioned above already, EVERIC Strong is one part of the modular concept contributing to more efficient processing.
Okay, but now one can easily say, let's make a vial as hard as possible. Like, does that make sense?
Yeah, so of course, you can try to make the glass as hard as possible. So you could, for example, toughen the glass. Yeah? The issue is that toughened glass could show even higher strength than certain materials that are used for the filling equipment of the filling line, and therefore could risk the damage of these parts. And this is nothing that we just invented spontaneously. There were really indeed partners approaching us, especially from machine vendors with this topic. And the second problem is that nothing is unbreakable. It is just simply a matter of the force that is applied. And the risk now with toughened glass is that if it breaks, it is bursting in many, many very small pieces. As consequence, this means the increased risk of glass particle contamination in your filling environment. And I think the third and most important point actually is this amount of strength is simply not needed. So our aim at SCHOTT was to achieve a reproducible strength that is meeting the requirements of a standard filling line and not over-engineering this product and with that actually introducing other even more difficult issues to solve with such a product.
Okay, this really makes sense to me, and I like the idea of balancing those different factors that influence, I would say, glass breakage and the strength of a glass. But we know that the glass strength is strongly influenced by two things: the flawless glass quality and different tensile loads that you just mentioned a little bit. Can you shed a bit more light on these points? How this could be affected?
Yeah, the thing is, you can have the perfect geometry, but then there's still the effect of cosmetics. The more glass to glass or glass to other hard materials contacts you have, the bigger is the risk that you introduce scratches, blunts, or bump checks. And these pre-damages in combination with applied loads, for example, during washing or stoppering on the fill and finish line, can then lead to glass breakage. On newer lines, a breakage is really a rare occasion, like already mentioned, even on bulk filling lines. But especially on older bulk filling lines, the handling of the vials is quite harsh, and this is leading to the introduction of multiple flaws.
Okay, you know I'm not an expert, Diana, on efficient processing, but just let me ask you a simple question. What if you could protect the outer glass surface?
Yeah. So this is exactly the initial idea behind EVERIC Smooth, protecting the outer glass surface during fill and finish from the introduction of flaws. And in addition, with EVERIC Smooth, we aimed at getting rid of the so-called sticking and climbing of vials, what often occurs in and after the deprotonation tunnel. And this behavior of vials is due to the fact that after washing and then heating the vials at approximately 300 degrees Celsius, the glass surface is highly reactive, and vials are most stress sensitive here and exhibit the highest friction in contact with other materials. So then the vials stick to each other and they climb at each other. And this can lead to glass breakage because those elevated vials might get stuck, for example, on the oven's exit. And this is what we wanted to solve with an outer coating, which has a decreased coefficient of friction. EVERIC Smooth.
Okay, I'm pretty sure that you will probably tell us more about it in your case study later on. But I would like to come back to your first line that you said, initial idea behind EVERIC Smooth. This sounds like there's more to tell. I assume this outer coating has an additional advantage.
Yes, it has indeed. And as you just mentioned, so last year we performed this comparative line trial at Hoffmann-La Roche's site in Kaiser-Augs in Switzerland. Yeah, and this was a great experience. So working in close cooperation with the operators and receiving this direct first-hand feedback. And during this trial, we noticed that EVERIC Smooth strongly decreases the cosmetic reject rate after final inspection. And this by a factor of almost 50 in comparison to the non-coated vials. And as you might know, the rejection of already filled vials is of course a huge cost factor, especially when you think of biologics, where one filled vial easily achieves a final price of around 1,000 euros. And you can imagine that this can be very beneficial, especially for a customer that offers a portfolio with a lot of high-value drugs, as also, of course, Roche is.
Wow, this is interesting because I like to have those real-life time production line tests, and it seems like that it is really beneficial what Roche and you found out. In a nutshell, so what are the three main findings in the Roche case study with EVERIC Smooth? Can you explain that a little bit to us?
Yes, so first of all, I think the main outcome is that with every smooth vials, the surface of the vial is protected. And this leads to the fact that the vial keeps its strength. And we proved this because we again did strength testing, and the results we received from the strength test really proved that that this is the case. And what we did, we draw samples of the EVERIC vials, and we draw samples of the standard vials at different locations of the filling line, and afterwards we tested burst pressure, axial and diametral load, in our lab again, and the results showed that the surface really protects the vial here. And secondly, I think with that trial, we showed that the coating is stable, because this is one worry our customers might have, that the coating is not stable. And what we showed is that there were no particles found related to the coating, neither in the filled medium nor in the washing water. And also washing and deprogenation didn't have an impact on the performance of the coating. And I think the third point would be that the coating, like already mentioned, majorly helps to decrease the cosmetic reject rate, which can heavily reduce your costs.
Yeah, you mentioned the 1,000 euros as an example for a market price for a biologics drug. This is fairly high, I assume. But have you made like a total cost of ownership calculation already? And how high are the potential savings when someone applies EVERIC Smooth to a glass vial? Is that something that you know about already, or even if it's in theory?
So, of course, this highly depends on various factors. The value of the drug that is filled, the existing yield performance, and therefore the potential for improvement, the quantity produced, and so forth. But let's assume, for example, a sales price of 1,000 euros per unit. And 3 million units that are filled per year. If we now take the 1.4% of yield improvement, what we achieved during the trial at Roche, this could easily lead to more than 30 million euros in savings per year.
Wow, this is amazing. Like, just from my understanding, wow, you said 1.4% of yield improvement that might lead to 30 million euros in savings per year. This sounds, at least from my perspective, this sounds to a very realistic case. So you told me in preparation of this podcast that you have presented this case study with reliable data to customers already. But besides those, I would say, clearly measurable results that you just elaborated on, what struck you most during your observations when you were in Kaiser Augst at the line test?
So one really surprising thing that I didn't have at all on my mind before was that it was much easier for the operators to pack the vials in the end. So the operators said that the vials kind of glided into the tray packaging. And also they said that pushing them into the washing machines was apparently much easier. And in addition, a lot of the operators also said, okay, it's so much less noise here. So we tested this as well, and indeed there was less noise. Also, they stated that the vials tremble less on the bells and that there is less wiggle when running empty. We also didn't see any breakage or climbing of the EVERIC vials during the trial. And what we and the operators also noticed is that after the tunnel, the standing vials were carried away much smoother by the moving vials from the rotary table. So no vial tipped over there. It's funny though, and I do not recall if we had the name already, so EVERIC Smooth, but the main message of the operators was they are running smoother. So apparently we decided for the right naming here.
This is funny because, but just what you all said, I'm like, this is something you can probably not plan in your R&D department, I think this is something you only find out if you do production line tests in real life. And this is something you probably did not expect at the beginning when you started to do this line test. During the course now of this podcast, you explained the two single features, EVERIC strong and smooth. But what are your thoughts on, because when I look at it, how you explained it, what are your thoughts on combining different features? And under what circumstances would you pick the one or the other combination? Is there something that you thought of already?
Yeah, yes, of course we did, because we always think in problems or in issues of our customers to then find the appropriate solution. And one example could be that you have a drug that is not sensitive, maybe even a small molecule, but you might have issues like sticking and climbing on your filling line, causing glass breakage, leading to downtimes. And in this case, just our outer coating, so just the module EVERIC Smooth would be enough and help you to solve that issue. And yeah, I think another example could be that you have a very sensitive drug in a ready-to-use packaging that will be filled nested. In this case, because there's no glass-to-glass contact and therefore no risk of introducing cosmetic flaws during fill and finish, a combination of EVERIC Pure and EVERIC Strong would be perfect. Yeah, and maybe a third example. So there you might have a sensitive, high-value biologic drug, for example, a monoclonal antibody, a huge and complex protein molecule, very costly from a production perspective. But this is filled on a bulk filling line, where you are facing the issue of throwing away products that have been rejected due to cosmetic defects after final inspection. And in this case, you could greatly benefit from a combination of all three modules, so EVERIC Pure, Strong, and Smooth. And I think that's a great offering for our customers. Choose that part of our EVERIC family that addresses your specific needs.
Wow. Okay. So thank you very much, Diana. We're running a little bit out of time now, but this was very interesting. And as I said it last week already, I always learn something new when talking to my product managers. So thank you so much, Diana, for sharing your expertise and insights with us and our audience here.
Yeah, Björn, and the only thing I can say is thank you for having me here, and thanks that I had this opportunity to share my experiences, especially with this line trial we had in cooperation with Roche. Yeah, because this was a really, really exciting experience. Thank you.
So I'm very happy that the internet line and connection was stable. So it was not interrupted in those times. And I'm pretty sure, Diana, we will hear each other again because I know that you have also other topics to discuss and to share your knowledge on that. I'm already looking forward to hearing you on one of our next podcasts. So I really like the modular approach of EVERIC that you just explained, especially because it creates a value product that matches with the requirements of the drug, and secondly, the expectations of our patients. So for everyone who wants to learn more about the physics behind glass and glass strength, I can only recommend our next podcast already. I met Dr. Florian Maurer, a well-known scientist in glass strength and fractography. He explains in simple ways how glass science works in the pharmaceutical industry. Until then, have a good rest of the week. See and hear you soon on one of our next podcasts at the Pharmaversity. Goodbye.